So this session will be called Actively Probing Connectivity in the Human Brain. And we're going to have three speakers, the first of which, Professor Dora Hermes. When I heard she was coming to Mayo, I heard she was an fMRI person. And as I've gotten to know her over the years, she's just a very good scientist that is able to do a lot of signal analysis. It's been an absolute pleasure working with her and having her as an asset to our group. Because I've learned a lot from her, and it's just been a really good symbiotic relationship for us to have her. She always gives excellent talks, and we're looking forward to this discussion this morning about algorithm development and network discovery. Thank you, Dr. Hermes.
We've really come from the era of localizing brain areas involved in disease to doing network-based discovery of biomarkers, diagnosis, and treatments. For example, you can see here the limbic network plotted with diffusion imaging in one of the patients who we work with. And this is just one of the networks in which we know that epilepsy can travel through these connections and affect different regions. And you can see, or for example, we know how Alzheimer's also can affect this network. But we want to do this in a way that we can not just apply this in one network, we can do network-based biomarkers and diagnosis and treatments across different networks. For example, those involved in the movement disorders, you've just seen addiction, or for example, mood disorders, etc. And I think if we think about the state where we're at right now, is we think we have maps. We know where these areas are located. We can map their function. We can even track the connectivity between these. But where we want to take this over the next years is to routing. We want to know if we have, for example, when we're going from Mayo Clinic, from the hospital to the clinics downtown, we want to know whether there's a road closure. We want to know whether there's a one-way street. We want to know how long it will take us to get there. And if we think about this development in terms of maps, from stationary maps to kind of dynamically evolving maps that indicate where there's a traffic jam, for example, and all these types of features, there's a lot of data that goes into that. For example, imagery data from satellites, street views, those are all ingested in these types of, to create these types of dynamic maps of the world. And for example, also third-party data providers and public data sources. So when we think about how do we create these routing maps for brains, we have a certain paradigm that we've started to explore, whether this works in this setting. And when we work with patients who have intracranial electrodes implanted during their stay in the hospital, we can work with them and, for example, and apply a very tiny pulse to one area and then see how this spreads across the network. And what we think what happens is when we stimulate one of these areas is that these pulses spread through the pathways in the brain, for example, the white matter pathways, as you can see here, and that after a certain time, they'll arrive at a connection site and maybe propagate further or maybe just stay there. And so during the single pulse stimulation, we can map networks and the information flow across those networks in the brain. And when we have those types of data, we want to make sure that we analyze those in a way that we can interpret it with respect to an individual, and we can actually discover these networks. And so together with our colleagues in neurosurgery, Kai Miller, we have developed several different frameworks in which we can assess these data to make sure that we have algorithms that are designed for discovering these networks. And the algorithms that we've, like the way in which we think about these is that we can look at hypothesis pre-selected areas over here, where we know that brain areas are connected through a certain set of bundles. And we know what type of pathway would connect them. And then we can see what type of electrophysiological biomarkers we have on each of these endpoints and how fast these networks are connected. How does information flow between these sites? We can also look in a divergent way, where we stimulate one site and we see how all those other sites respond to stimulation in that one area, to see when we stimulate an area, how does it drive the rest of the brain. We can also look in a convergent setting, which is really important when you're starting to think about a disease affecting a certain set of areas. What are the potential targets that you could stimulate to drive this single area? And so for each of these paradigms, we've proposed different types of analyses that optimize the discovery in those networks. For example, to optimally characterize the responses, which we call canonical response parameterization, which work really well for the hypothesis pre-selected and the divergent paradigm. When we look convergently, we've developed a technique called basis profile curve identification that can better understand how all these different areas drive a single area and what we can then learn from these different types of inputs. When we do this work, we don't only want to have paradigms or algorithms that analyze these data in an optimal fashion, we also want to have the data be able to ingest those data as automatically as possible in those algorithms. So we've developed or contributed to several data science tools. One of those is the brain imaging data structure, which is a machine-readable standard where you can harmonize data in a standardized way, both the primary data, but also the metadata, such that however I store my sampling frequency is going to be the same as however MEG or EEG stores their sampling frequency. And we don't have to search for this term. We know what it's called. It's not one person calling this SF and another person calling this sampling underscore frequency. We know what it's called, and we can find it in a machine-readable fashion and feed it into our algorithms in an automated way. So we contributed here to only a small portion. We contributed to the IEG BIDS extension, but you can see that this brain imaging data structure is also being developed by an international community for MEG, EEG, genetics, ASL, PET. Started out with MRI, but also branching into all these other different frameworks. And so when we have these things in place, when we have the algorithms in place, when we have the data science in place and the paradigm, we can then start asking questions, interesting questions, like what's the typical transmission speed along the superior longitudinal fasciculus? And this is something that, we may think we know, like, don't we know this already? Isn't this something that's already been figured out in the 1980s? And it's actually a very interesting question, because when we started to look at this, it's actually difficult to notice in human brains, because human brains are unique compared to many other primates. What you can see here on the left is that in human brains, we have this additional stage of myelination, what you see on the y-axis. After the age of 20, you can see this additional growth of myelin, and that in the chimpanzee, you don't see that. So when we actually ask these questions, like how fast do your signals go, we actually need human measurements, because we can't just generalize this from our non-human experiments. And so how long does it take to get from parietal to frontal areas? We started measuring these evoked responses, stimulate parietally and measure frontally, and then measure the latency. And we can see, and then we ordered this as a function of age, between age of 10 and 50 years old. And you can see that a 10-year-old has a latency around 20 milliseconds, and a 30 or 40-year-old actually has a latency around 50 milliseconds. And we can see that this actually is a growth curve, and we can see that we're still developing up until the age of about 40 years old. And what happens after that, we actually don't know yet, because we didn't have sufficient older patients in this cohort. So we can just assume that it stays stable at that age. We can do the same for other pathways, such as the arcuate fasciculus. We can see here that the decrease in latency is similar compared to the superior longitudinal fasciculus. And we can see that this decreases with development, and these type of developmental curves actually match what we see based on imaging, is that there's this long developmental timescale, and a 20-year-old is not an adult yet, even though they may try to convince you otherwise. And so when we start to think about this, we know the latencies. We can now calculate these speeds, right? How fast can we drive across those pathways? How fast are those pathways? Are they like, is it like a highway, or is it more like a bike path, or is it more like a muddy road? How fast can we go? And so we can calculate the distance between these sites. And when we have the distance and the latency, we can calculate what the average speed would be. And we can see that we increase from about a speed of about 2 meters per second, which is the speed of about a person walking. And when you're like 30 or 40 years old, you must be super happy, because you can actually go at the speed of biking. which we've heard before actually makes me very happy because I'm from the Netherlands. And so when we're starting to be able to calculate these speeds, we can see how our brain progresses and how we need individualized biomarkers for patients of different ages. But that's not the only thing we can calculate with these type of measurements. When we stimulate here, we don't only record in these direct connections, sometimes we also record in these indirect connections. And this is really interesting because it might tell us something about the areas in between and about the excitability, like how do these signals propagate across synapses to other areas. And one of the things that we started to notice when Dr. Greg and I were recording these data in the ICU, and when a research tech, Gabriela Ojeda Valencia, was looking at all these images, all these data coming in, we started to note that when we stimulate the hippocampus, and we measured in the posterior cingulate, that in every individual trial, we can see that there is this peak in the signal around 200 milliseconds, which is a very, very long time in terms of brain dynamics. It cannot be a direct pathway. So what is potentially going on here? And so what we thought about when we map when we follow this known anatomy, we integrate it with the imaging in an individual patient. We know that we're stimulating the hippocampus. We know that we're measuring in the posterior cingulate. It's very unlikely to be governed by this direct pathway because this red pathway is too posterior. It doesn't actually end in where we're measuring. So it could also be, alternatively, this pathway through the fornix, the green line, to the mammillary bodies, to the mammillothalamic tract, to the ANT, and then from ANT through the blue tract to the posterior cingulate. So we also stimulated the ANT, and what you see when we stimulate ANT, what we then measure in the posterior cingulate is a response that is just 40 milliseconds earlier, but very similar in waveform. And so what we infer here is that when we stimulate the hippocampus, it travels through this indirect pathway, and which is interesting because then this type of characteristic waveform may tell us something about what's going on in this network, and we can use it as a biomarker to identify or to see how these networks change in diseases like epilepsy. And so the last thing I would like to show is how we can take this from this hypothesis pre-selected framework to a divergent framework. What happens if we stimulate these different areas? What are the projection zones of influence? And this is very interesting to test in type of these thalamic sites, such as the pulvinar, because the pulvinar is one of these new sites that's becoming a stimulation target to modulate visual pathways in epilepsy. In particular, when there's several sets of studies that have shown in preliminary results from clinical trials or case reports that show that this is actually effective and this can help patients. And from one of these reports, you can see, however, that these electrodes are all placed in the pulvinar, but in pretty different areas of the pulvinar. And now the pulvinar is relatively large. It's a relatively large nucleus with different functional and structural connectivity areas. For example, the lateral pulvinar, here circled in red, having connectivity to early visual and extrastriate areas. The posterior medial pulvinar in green having connectivity to parietal and frontal areas. And the middle pulvinar, ventral medial pulvinar, having more connectivity to middle temporal areas. So our question was here is, can we map this type of detailed connectivity in an individual such that you can optimize the implant sites if seizures come from middle temporal or parietal or frontal or early visual areas? So we looked at a lot of the different patients we'd seen over the last year since they started implanting electrodes. These wonderful neurosurgeons started putting electrodes in the pulvinar, and we tested what the different output pathways would be. And what we would see if we look at the medial sites and we stimulate lateral pulvinar, we see this strong output in early visual areas. Whereas we don't see that when we stimulate ventral or dorsal medial pulvinar. When we stimulate the ventral medial pulvinar, we see strong outputs to lateral temporal areas that are much weaker in these other two cases. When we look at the interparietal sulcus here on the bottom, we see that it gets strong inputs from dorsal medial pulvinar that we do not see in these other two cases. And this is also in an individual patient. You can see how close those electrodes are together here in the pulvinar. And we can see that here on the bottom, when we stimulate the lateral pulvinar site, there's beautiful outputs to these early visual areas. And we see that we are missing that when we're stimulating the ventral medial pulvinar. If we look at these lateral temporal areas, we see strong inputs from this ventral medial pulvinar site, and we miss those entirely if we go to the lateral pulvinar. And so this is very important. And also what we saw in this case is that, for example, we do not see any outputs in the hippocampus, which has been proposed from some previous fMRI studies that has been connected to the pulvinar. But when you look at more detail, these pathways likely go from the hippocampus to the pulvinar, but not from the pulvinar to the hippocampus. So not all these pathways are bidirectional, and we have to take that flow into account, and we can measure that in this way. So to summarize, we can use this type of single pulse stimulation to combined with network-targeted computational frameworks to discover neural information routes that are strongly linked to anatomy. We can discover the speed of these networks. We can discover biomarkers for indirect and for direct routes. We can look at outgoing influence and also at incoming influence and discover these one-way routes that are important in individual patients if you want to modulate an area. And we combine this with data science for multimodal data, so that we can characterize networks in individual patients, and that we can start comparing upcoming patients also to previous patients that we've seen. And we can compare patients with different types of epilepsy, for example, or different types of diseases. So we can compare across individuals. And so I think at this stage, we're starting to be ready for this type of clinical application, and I'm really excited to see the next talk from our wonderful colleague, Dr. Greg. So thank you so much. I would like to thank you. And, thanks for